told you the most important discovery in biological history wasn't carefully planned, but stumbled on through rivalry, secrecy, and a photograph almost nobody saw. Today on Knowledge Forge, we're going to take a look at the story behind the discovery of the structure of DNA. Everything about our understanding of biology in the modern world and all the medical miracles that have come since can be traced back to the discovery of DNA. By the early 1900s, scientists knew that traits were inherited. Eye color, height, disease, all of these were easily traced along familial lines, and the fact that traits were passed from parent to offspring was well known, but no one really knew how. Most scientists believed proteins carried heredity. They were complex and varied and came in dozens of types. DNA was thought to be a little bit too simple, just 4 repeating letters, A, T, C, and G, and it seemed impossible that something so basic could encode life. But they were wrong. In 1944, the Avery-McCarty-McLeod experiment conclusively proved that DNA was responsible for carrying genetic information when the scientists were able to successfully isolate DNA from protein in pneumonia-causing bacteria. Through a series of chemical tests, they were able to put the protein versus DNA debate to bed once and for all. In the 1950s, labs across the world raced to solve the DNA structure in hope that understanding the chemical nature of the molecule might help scientists make sense of how such a seemingly simple compound could result in almost infinite genetic variation. At Cambridge, the team was James Watson and Francis Crick. They were brilliant, impatient theorists with a bit of a reputation as lazy jokers. Meanwhile, at King's College, the team was Rosalind Franklin and Maurice Wilkins. Franklin was meticulous, methodical, experimental, but she lacked the respect from her male colleagues, including Wilkins. Watson and Crick spent their time trying to attack the problem from the chemical end. They built models of how the chemical bonds might orient themselves in a way that could store information, but that method resulted in little success. In fact, when they tried to share their early model with the King's College team, Rosalind Franklin laughed at them because it was so clearly wrong. The King's College researchers, on the other hand, used a technology called X-ray crystallography to try and photograph the DNA itself, and in 1952, Rosalind Franklin captured something extraordinary. The image was called Photo 51. It was a blurry, X-shaped pattern, but to trained eyes, it was unmistakable. An X means a helix. The spacing means 2 strands, and the angles revealed its dimensions. That single image contained the answer, but Franklin wasn't credited for what she'd captured. You see, at the time, there was some confusion about who the head of the X-ray crystallography project was at King's College. Wilkins was the one who originally started the program, and he believed himself to be in charge of the experimentation, and was rather cold towards Franklin. He viewed her as an outsider that the university had put there to babysit him. As a result, she was less than forthcoming with her own findings and kept Photo 51 a secret, hidden in her office where she attempted to work out the problem on her own. It was mostly down to chance that one day James Watson made the trip from Cambridge to King's to try and draw inspiration from Wilkins. He found himself in Franklin's office alone, and her groundbreaking research was in clear view on her desk. Watson immediately sketched out a copy of the image and hurried back to Cambridge to confer with Crick, who was rather good at interpreting crystallography images. Within weeks, they had built the world's first double helix model of DNA. In 1953, the structure of DNA was published. Watson, Crick, and Wilkins were each awarded with the Nobel Prize in 1962 for the discovery. Franklin passed away in 1958 and was ineligible to receive the prize because it can't be awarded posthumously. She never knew that it was her research that had proved the key to allow Watson and Crick to finally unlock the secret of DNA. There were 2 strands, twisting, each letter pairing perfectly, A with T, C with G. Suddenly, heredity made sense. DNA could copy itself, and errors could create variation. Life could change and evolve. Biology was no longer descriptive, it was mechanical. DNA reshaped medicine, forensics, ancestry, and evolution. It allowed us to diagnose diseases, trace crime, and uncover our origins. But it also revealed something humbling. Every human shares over 99.9% of the same DNA. We're all just variations on the same theme. The discovery of DNA wasn't just scientific, it was human. It was full of brilliance and conflict and pettiness and unfinished credit. It changed humanity forever, and It's still really important to this day. Thank you for watching. Hey, you made it to the end. Thanks for that. If you enjoyed this video, you might want to check out this one about how we decoded the human genome, and you can click over here somewhere to subscribe to Knowledge Forge so that you never miss an episode. Thanks for watching.